The brain is one of, if not the most important organ of the body. It serves as the central command centre of the nervous system, responsible for controlling and coordinating all bodily functions, thoughts, emotions, and behaviours. For this reason, the brain has several inbuilt structures that function to protect it. The first protective structure or structures of the brain are the protective coverings. These include the bones of the skull and the meninges. The meninges are three protective connective tissue coverings that encircle both the brain and the spinal cord. And from superficial to deep, or the outermost to the innermost layer, these are the dura mater, the arachnoid mater, and the pia mater. The dura mater is the most superficial of the meninges and is made up of two layers of thick, strong, dense, irregular connective tissue. It's represented by this green line along here. Around the brain, these two layers are fused, except where they separate to enclose the dural venous sinuses, which are endothelial-lined venous channels that drain venous blood, or the blood in the veins, deliver it to the internal jugular veins where it eventually returns to the heart. So this is one of our venous sinuses here. The arachnoid mater is the middle layer of the meninges. It's represented by this pinky purple line along here. It's a thin avascular layer, so avascular meaning free of blood vessels, and is composed of loosely arranged collagen and elastic fibres. It's called the arachnoid mater, with arachnide relating to spider, because of its spiderweb-like arrangement of those protein fibres. Between the dura mater and the arachnoid mater is a thin space called the subdural space, which contains a small amount of interstitial fluid. And finger-like extensions of the arachnoid mater, called the arachnoid villi, which are these pink structures here, project into our venous sinuses and work to gradually reabsorb cerebrospinal fluid back into the blood. The pia mater is our innermost layer of the meninges and is a thin, transparent connective tissue layer that adheres directly to the surface of the brain and the spinal cord. So it moves in and out of all of the grooves of the brain. It consists of thin squamous to cuboidal cells with interlacing bundles of collagen fibres and some elastic fibres. And within the pia mater are many blood vessels that supply oxygen and nutrients to the underlying nervous tissue. Between the arachnoid mater and the pia mater, so the middle layer and the innermost layer, we have a space called the subarachnoid space, which contains our cerebrospinal fluid, which we're going to come to shortly. So recapping that in text for you, so you don't have to make too many notes, the meninges are three protective connective tissue coverings that encircle both the brain and the spinal cord. From superficial to deep, they are the dura mater, the arachnoid mater, and the pia mater. So the dura mater is our outermost layer, made up of that thick, dense, irregular connective tissue. It encloses our dural venous sinuses, which are those channels which drain the blood from the brain. The arachnoid mater is our middle avascular layer of thin, loosely arranged collagen and elastic fibres that looks a lot like a spider web. Projections from this arachnoid mater, which are called arachnoid villi, project into those venous sinuses and reabsorb our cerebrospinal fluid. The pia mater is the innermost layer of those thin squamous to cuboidal cells with collagen and elastic fibres. It adheres directly to the brain tissue, and it contains blood vessels which supply oxygen and nutrients to that underlying brain tissue. The next protective structure we're going to take a look at is the blood-brain barrier. And the blood-brain barrier is a term used to describe the unique properties of the blood vessels of the brain. So it's not necessarily a particular structure, but a description of the unique characteristics of the blood vessels of the brain. Now, just as a bit of background, despite the brain representing only 2% of body weight in adults, it receives between 15 and 20% of the total volume of blood pumped around the body. And even at rest, it's thought to consume about 20% of the oxygen and glucose used by the entire body. Now, neurons, which remember are our nerve cells or our brain cells, produce ATP almost exclusively from glucose via reactions that need oxygen. So when the activity of neurons or neuroglia increases in a particular region of the brain, blood flow to that area also has to increase the delivery of oxygen and glucose to those tissues. So the blood-brain barrier is made up of tight junctions that seal together the endothelial cells of the capillaries that feed the brain. A tight junction just means that the endothelial cells surrounding the capillary meet really tightly, rather than having a small gap between cells, which is what we see in other blood vessels in other parts of the body. So these yellow cells here are our endothelial cells. You can see these cells join really tightly. Those cells are overlapping. In a typical cell elsewhere in the body, we'd actually have quite a decent sized gap between those cells because our capillaries are quite leaky or highly permeable, which means that substances can go from the bloodstream out into the tissues quite easily. Now, in the brain, in the blood-brain barrier, we also have a thick basement membrane surrounding those capillaries. And then surrounding that basement membrane are the processes or the end feet of many astrocytes, which remember is one of our neuroglia within the central nervous system. And these press up against the capillaries to form another layer of this boundary, as well as secrete chemicals that are thought to maintain that permeability or the tightness of those tight junctions. So you can see around this capillary, we really have 1, 2, 3 layers. Now, the function of the blood-brain barrier is to, as the name suggests, act as a barrier between the bloodstream and the brain tissue, allowing only certain substances, so things like water, oxygen, some small lipid-soluble molecules to actually pass through, and then prevent any toxins, pathogens, and other potentially dangerous substances from crossing from the blood into that brain. Trauma, certain toxins, and inflammation can all cause a breakdown of the blood-brain barrier, which increases the permeability, so it opens up these tight junctions and therefore increases the risk of a potentially harmful substance moving from the blood into that brain or nervous tissue. Now, this is not specifically a part of the blood-brain barrier, but it is a part of the circulatory system which supplies the brain. And this structure or diagram I have up here is called the Circle of Willis. And the Circle of Willis is a circular arrangement of interconnected arteries that sits at the base of the brain. It works to essentially provide a backup route for these arteries to ensure a continuous supply of blood to the brain. So this circle, which you can kind of see here, and it's a loose use of the term circle, is formed by the convergence or the meeting of several major arteries, and it helps distribute blood flow to many different regions of the brain, particularly in the event of a partial blockage or a disruption in one of the contributing arteries. So, for example, if we take this vessel here, if there's a blockage here, the blood can either go this way or it can go around this way to reach its target, reach where it wants to go. If this was anywhere else in the body and we have a typical arrangement of blood vessels where it's just one line, if a blockage is here, anything downstream of that blockage would simply go without blood. So this circular arrangement is just basically a backup route for the blood to take in the case of a disruption or some kind of blockage. So again, popping that information into words for you, our blood-brain barrier is made up of endothelial cells with tight junctions, a thick basement membrane, and the end feet of the astrocytes, which surround the capillaries of the brain. It works to separate the brain tissue from any potentially harmful substances in the blood. The circle of Willis is a circular arrangement of interconnected arteries at the base of the brain, which works to ensure a continuous supply of blood to the brain, even in the event of a blockage or a disruption in one of the involved arteries. The last protective structure we will cover in this module is the cerebrospinal fluid. You may see this referred to as just CSF. And the cerebrospinal fluid is a clear, colourless liquid composed primarily of water that protects the brain and the spinal cord from both chemical and physical injury. So cerebrospinal fluid continuously circulates through cavities of both the brain and the spinal cord, as well as around the outsides of the brain and the spinal cord in the subarachnoid space, or that space between the arachnoid mater and the pia mater. The total volume of cerebrospinal fluid in an adult is between 80 and 150 millilitres, so not a great amount. And what you can see on this slide here are the 4 continuous cavities, which is where our cerebrospinal fluid is produced and through which they flow through. So these blue cavities within the brain are called the ventricles. There are 2 lateral ventricles, so one in either hemisphere or the left and the right side of the brain. These 2 actually meet in the middle, but they are separated by a thin membrane, so they are 2 separate cavities. The 3rd ventricle is a narrow slit-like cavity that runs along the midline of the brain and right between the left and the right halves of the thalamus, it's like a circular structure in the middle of the brain, which again we'll talk about shortly. The 4th ventricle then lies between the brainstem and the cerebellum. So this is our brainstem here, this is the cerebellum. And it's continuous with the central canal, or the hole that runs down the centre of the spinal cord. So these cavities are where our cerebrospinal fluid is produced and where they will flow through. So looking at those ventricles at another angle, we're now looking down on the brain in this image here. You can see these are our 1, 2 lateral ventricles here. This is the septum pellucidum, which is that thin membrane that separates those 2 ventricles. And as I said, our cerebrospinal fluid is produced in our ventricles through a structure called the choroid plexus. So the choroid plexus are just a network of blood capillaries which sit in the walls of the ventricles. Taking a closer look at that choroid plexus in this image up here, and a choroid plexus is made up of ependymal cells with really tight junctions which cover that network of capillaries. So selected substances that are found in the blood, so this is mainly water, will be filtered through those tight junctions of the ependymal cells. What ends up out in the ventricles is then our cerebrospinal fluid. So it's nice and tight, has those nice tight junctions, so only selected substances will actually move into the cerebrospinal fluid. Now, the movement of substances across those ependymal cells doesn't actually just only go from the blood into the ventricles, it can work the other way. So we can have substances go from the cerebrospinal fluid and be returned back into the blood. So just to show you the pathway of the cerebrospinal fluid through the brain, we have our 2 lateral ventricles up here where we start to form our cerebrospinal fluid with those choroid plexuses. They flow down through into our 3rd ventricle where we add more cerebrospinal fluid, flows down through our cerebral aqueduct into our 4th ventricle where again we produce more cerebrospinal fluid. That flows down the central canal of the spinal cord, so that hole in the middle. It then moves into our subarachnoid space, so that space between the arachnoid mater and the pia mater, circles around the outside of our spinal column, spinal cord, and then around the outside of the brain, where it returns to this venous sinuses and it gets reabsorbed by that arachnoid villi. So cerebrospinal fluid has 3 ways in which it protects the brain and the spinal cord. It serves as a mechanical protection, so a bit like a cushion. It absorbs shock and protects the delicate tissues of the brain and the spinal cord from any jolts or movements that would otherwise cause them to hit the bony walls of the skull and the bony walls of the vertebra. The fluid also allows the brain to float within the skull, so that the brain is actually reasonably heavy. We don't want that delicate tissue just resting on the bottom of the skull. So by having this cerebrospinal fluid, it can actually float within the skull and not damage that tissue on the bottom of the brain. It also has a homeostatic function whereby it can regulate its own pH. This has subsequent effects on pulmonary ventilation or breathing and blood flow. It can also serve as a transport system for hormones within the brain. Lastly, cerebrospinal fluid also plays a role in circulation by acting as a site of exchange for nutrients and wastes between the blood and adjacent nervous tissue. The cerebrospinal fluid can also adjust intracranial pressure, so that's the pressure within the skull, to facilitate blood perfusion, which is essentially just blood flow. So if we increase the production of cerebrospinal fluid, you have more fluid within the skull. This increases intracranial pressure, and that actually inhibits blood flow. Going the other way, if we decrease the amount of cerebrospinal fluid we are producing, there is less volume of that fluid within the skull,